This is our DNA replication model video. DNA, or deoxyribonucleic acid, is a double helix molecule which carries genetic information. DNA is made up of nucleotides. Each nucleotide contains a phosphate group and deoxyribose, in addition to a nitrogenous base. In this drawing, the phosphate groups are represented by circles labeled with the letter P. The two strands are anti-parallel, which means that one goes from 5' to 3' and the other 3' to 5'. The deoxyribose sugars are represented by pentagons because deoxyribose is a 5-carbon sugar. There are 4 different nitrogenous bases in DNA: adenine, thymine, guanine, and cytosine. Adenine pairs with thymine, while guanine pairs with cytosine. The pairs are binded through hydrogen bonds. These nitrogenous bases are represented in this drawing with the letters T, A, G, and C, respectively. Now that we know the structure of DNA, let's go over DNA replication. DNA replication first begins at areas called origins of replication, which are short stretches of DNA with a specific sequence of nucleotides. First, proteins called helicases attach to the DNA and untwist the strands at the replication fork, as represented by the green marker. Once the strands separate, single-strand binding proteins, or SSBPs, bind to them, which keeps the strands from repairing. Next, primase synthesizes an RNA primer at the 5' end of the leading strand, the strand to the right. Now, our DNA polymerase III is able to use parental DNA as a template and synthesize a new DNA strand by adding nucleotides to the previously created RNA primer. The lagging strand, which is the strand to the left, works a little bit differently. Primase synthesizes a RNA primer at the 5' end of each Okazaki fragment of this strand. DNA polymerase III is now able to use the parental DNA as a template and synthesize a new DNA strand by adding nucleotides to the previously synthesized RNA primers at each Okazaki fragment. DNA polymerase I then removes RNA nucleotides from the 5' end of one of the fragments and replaces them with DNA nucleotides. Lastly, DNA ligase joins the Okazaki fragments of the lagging strand. Next, we are going to talk about mutations. The first type of mutation is substitution. In this instance, a nucleotide pair is substituted for another. This changes the mRNA sequence. There are 3 different outcomes: a silent mutation, a missense mutation, and a nonsense mutation. During a silent mutation, there is no effect on the amino acid sequence. However, during a missense mutation, there is an effect on one amino acid usually. And for a nonsense mutation, a codon is substituted for a stop codon. The second type is insertion. During this case, a nucleotide pair is inserted. This signifies a frameshift mutation, which causes immediate nonsense because the extra nucleotide pair affects the original amino acid sequence. The last type is deletion. This occurs when a nucleotide pair is deleted from the sequence. This can cause frameshift or 3 nucleotide pair deletion.